I'm going to talk about the evolution of recombination today, but I'll start by giving a kind of broader introduction. I first want to mention the folks who have done the work I'm going to talk about today. April Peterson was a PhD student in the lab. Richard Wang was a PhD student in the lab. Actually, all of these people are graduate students, or were graduate students when they did this work. Beth Dumont is now a professor at the Jackson Lab, the sort of capital of mouse genetics. Emma Howell is currently in the lab. But I'm very grateful to all of these students for the tremendous effort they put into these studies that I'll describe in a moment. Okay, so I think that most of you are probably familiar with meiotic recombination, but just to make sure we're all on the same page, recombination is this somewhat peculiar process that happens during a prophase of the first meiotic division. So remember, we have two meiotic divisions, and during the beginning of the first one, cells do something that seems kind of strange. They induce damage to their own DNA. They form double-strand breaks in a programmed manner, and some minority of those double-strand breaks all have to be repaired, of course, and the cell has machinery for doing that. But it's a minority of the double-strand breaks that are repaired as crossovers, so that you get the products coming out of meiosis 1 have swapped DNA between the parental and the maternal chromosomes, and then that's carried into meiosis 2. So this is a general process of great importance to cells and organisms. And one of the reasons it's so important is that, for reasons that are not fully understood, recombination is required for proper segregation of chromosomes in most species. So errors in recombination can produce birth defects that arise through aneuploid gametes. For this crowd, you might be at least as interested in the fact that recombination diversifies offspring genomes. It's one of the forces that creates unique genetic combinations, so that siblings are so that siblings look and act different from one another. And in the process, it shapes evolution. And in fact, recombination is a basic parameter for population genetics that is included in all population genetic models that involve sex. So just to go in a little bit more detail, when we start with chromosomes, and a mutation arises or exists on some subset of chromosomes, then it's recombination that determines the scale of non-random associations with other mutations or linkage disequilibrium. Namely, if we have a high recombination rate surrounding that mutation, or many generations of recombination, it erodes linkage disequilibrium so that the scale of associations is short. In contrast, if we have a low recombination rate or few generations of crossing over, we end up with long haplotype blocks, lots of linkage disequilibrium. So recombination shuffles combinations of alleles, it erodes linkage. Another more applied way to think about recombination is when we construct population genetic models, usually the processes of interest can be detected or analyzed by thinking about how they distort gene genealogies. And recombination shapes variation by altering gene genealogies. As you look backward in time, if you have crossovers like those depicted by the dashed lines in this figure, it actually changes the genealogy and structures how it alternates along the chromosome. So it's a basic parameter that we need to keep in mind when analyzing population genetic data. Now, what you might not know is that, when we do, for example, coalescent simulations, you tend to input like some fixed recombination rate. And I understand why we do that, but the reality is that the recombination rate is not fixed. In fact, it evolves. This is a summary from a review paper. Notice that on the y-axis, it's a log scale of recombination rate. So centimorgans per megabase. This means centimorgans tells you, 1 centimorgan, it means that 1% of the offspring are going to be recombinant for 2 markers that are 1 centimorgan apart. And we often divide by megabases just to standardize them. But what you can see here is that there's remarkable variation in the meiotic recombination rate, which is sort of intriguing because again, this is a process that's required in most species for meiosis to complete properly, and you might expect there would be stronger constraints, but the recombination rate evolves. And it varies not just between species, but also within species, as I'll show in a minute. So people have gotten excited about studying the evolution of recombination rate. They've actually been doing it experimentally for decades, but recently advances in genomics have further encouraged people or excited people to look at how recombination rate evolves. And from where I sit, despite this ramping up of interest, there are still some key unanswered questions that I'm going to try to address today. What are the genetic determinants of recombination rate evolution between species and between populations? Does recombination rate evolve separately in females and males? And finally, I'm going to show you a little bit of data and speculate about this last question, which is the hardest one for us to answer. How might natural selection shape recombination rate evolution? Okay, so I first want to, before I start showing you some experiments and some results that we think are exciting, I want to just impress upon you that there isn't just one recombination rate. Like any rate, it depends on the scale over which you measure it. So on the finest scale, at the top of the figure here, we can imagine that there's heterogeneity in the rate of crossing over along a chromosome that can be really fine, really rapid changes in the recombination rate. In some species, including mammals, there are so-called recombination hotspots where a disproportionate number of crossovers occurs. And they're surrounded by recombination cold spots. And then kind of one up from that we could imagine collecting crossovers or counting crossovers in intervals that might be megabases in size. And we see more gradual transitions in the recombination rate at that level. And then at the level that is probably most important for fitness is the whole genome level, where we count crossovers that happen on each chromosome and sum them together. And I say it's most important for fitness at this level because what is needed for meiosis to complete successfully is usually at least one crossover per chromosome. And there may be some upper limit after which the DNA damage that's incurred becomes hard to repair. So today I'm going to focus mostly on this last one, the evolution of the whole genome recombination rate as a kind of cellular readout. But towards the end, I'll talk a little bit about the fine scale rate. All right, in my lab, we use house mice as a model system for studying evolution, whether it be giant mice living on islands, as Christian mentioned, or thinking about how new species arise. And we also use them to understand recombination rate evolution. They have many advantages. One is that we have inbred strains from around the world, and we can take replicate recombination rate measurements using genetically identical mice to try to understand the genetic basis of recombination rate. We can also compare variation within and between groups across multiple recent timescales because of the radiation of house mice. And although we're chiefly interested in what happens in mice as a model for organisms in general, it's nice that some of the findings that arise from studying house mice may be relevant to recombination mechanisms in humans. House mice are organized into three principal subspecies. So the binomen is Mus domesticus, and we've got three subspecies. Mus domesticus domesticus is the Western European subspecies. Mus domesticus, sorry, it's funny, I'm naming the wrong species. It's Mus musculus domesticus. Mus musculus is the Eastern European and Asian subspecies, Northern and Central Asian, and then Castaneus is the subspecies mostly from the Southeast. And since you live in Vienna, you may know about the famous hybrid zone that runs through Germany and Austria, where domesticus and musculus meet and exchange genes. But these are the three subspecies that we study. And we have an opportunity again to look at evolution of any trait between subspecies. This is a rough depiction of the phylogeny of the three subspecies. There's incomplete lineage sorting and things, but this is basically what it looks like. We can make comparisons here, but we can also make comparisons within subspecies. So this is quite helpful. Okay, so how do we actually study recombination? Well, because we're interested in the genetics of differences in recombination rate, we need a method to estimate the recombination rate in individuals. So we don't want to try to generate a genetic map for each mouse. So instead, we're using an immunocytological approach. And it takes advantage of the following facts. This is a picture showing what's happening in meiotic prophase, the prophase of the first meiotic division. And you can see the sister chromatids are labeled with loops, and they're so each of the kind of rings here and the loops that go with them is a homologue, a homologous chromosome. So one of them is from mom, one of them is from dad, and then they're tethered together by these green dots that form something called the synaptonemal complex. So this, you can think of this as the proteinaceous glue between the two homologues that allows them to stay together through that, allows them to stay together while crossing over is happening. And this big fluorescent oval represents a focus of an MLH1 protein, which is a DNA repair enzyme that shows up and repairs double-strand breaks into crossovers. So this is really helpful because if we can look for MLH1 foci, we can count crossovers in individual cells and in individuals. And that's what we do. So here are some actual pictures from mice. A spermatocyte from a male, 5 to 12 weeks of age, when we can get access to spermatocytes that are undergoing recombination on top. And at the bottom here in B, we see a female, an oocyte, and this, to get access to recombination in females, we need to sample them either late on as embryos or less than a day after they're born. And what you can see is there are these red lines, and the red lines are the synaptonemal complex. So you have to kind of imagine on either side of the red line is a homolog that is not being visualized. And the green dots are MLH1 foci that represent crossovers. So we can literally count the number of green dots that localize with synaptonemal complexes, and that tells us how many crossovers have happened. And this is what I just said. So the green dots are the crossover sites. What we've done here is to stain with antibodies. Okay, so now we're in a position to address this first question, which is going to be a big chunk of my talk today. Because we can measure recombination rates across the genome, counting the number of crossovers that happen in each individual, we can use standard genetic mapping to look for regions of the genome associated with recombination rate. And Beth Dumont did this several years ago. She started with an inbred strain from the subspecies musculus and crossed it to an inbred strain from the subspecies castaneus. And she did this cross because, as you'll see in a minute, these two strains differ in the total number of crossovers by more than 30%, which is a startling result that Beth first found. So she crossed these inbred strains to construct F1s, which are genetically identical on the autosomes, and then crossed the F1s to generate a large panel of F2s. And she measured, using this immunocytology approach I just mentioned, she counted crossovers in every one of these 315 F2 males. She focused on males here because even though we've collected data for recombination in females, as you'll see later in the talk, it's a lot harder to come by. So she's looking at divergence in the male recombination rate and trying to map it here. Okay, so this is what the distribution of the total number of crossovers in a cell looks like across all of these 315 F2 males. So I first want to orient you to the dotted lines that show the mean number of crossovers for the musculus strain from the Czech Republic, which is about 30, and the mean for castaneus, which is about 22. So this is quite a huge difference. And when you look at the F2s, you can see that they span the range. It looks like sort of a standard quantitative trait in the sense that there's a continuous distribution that is mostly encompassed by the two parental strain means, but there are some individuals that exceed those values. So this is a great sort of distribution to do genetic mapping. And that's what Beth did. So she genotyped the 315 F2s at a bunch of informative markers. And what she discovered is that this difference in recombination rate that is probably between subspecies is in part caused by 8QTL with a whopper on the X. So each of these bumps is a location on a particular chromosome that contributes or affects the genome-wide rate of crossing over on the autosomes. So the number of crossovers on the autosomes. And this was our first look at the genetic architecture of this trait. And I'll just point out that this whopper on the X explains 35% of F2 variance. And for those of you who are interested in quantitative genetics, you'll recognize that that's a huge effect size. But there's a twist associated with that quantitative trait locus that I'm going to mention right now. So this table, I just want to highlight a couple of things. This is summarizing the effects on recombination rate of each of these QTL. And multiple QTL mapping gave us kind of the best portrait. That's where the 8 came from. So if we look at those lines, those rows, it's helpful. But I just want to point out that if you look at the autosomal QTL, we see a fairly consistent pattern that the allele descending from the low parent, the parent with the low recombination rate, which is cast, has the lower is associated with the lower recombination rate in the F2s. This makes sense, right? So the musculus or PWD allele, which is the high parent, increases recombination rate for all of the autosomal QTL and for one of the X-linked QTL. But when you look at this box, this blue box down here, you can see that that's not true for the QTL that explains 35% of the variance on the X. It has so-called antagonistic effects where the high parent, the allele from the high parent, is actually associated with a lower recombination rate. So this is our first clue that the effects of QTL on the recombination rate are not going to be simple. Okay, so enter Richard Wang, who did a similar kind of project, but now looking within subspecies, looking at crosses involving strains from two populations. One of them is on Gough Island, where the giant mice roam, and another one is from a population in Maryland in the United States. And so Richard did the same kind of, used the same kind of approach. He generated F1s from these inbred strains and then constructed genetically unique F2s, and he got up to 240 F2 males. And he scored the number of crossovers using the approach I mentioned before in each of these males. Here's what the distribution among the F2s looks like. In this case, the difference in the total recombination rate between Gough Island mice and this other mainland strain, WSB, was much smaller than the difference between subspecies that Beth had analyzed. And so a lot of the F2 individuals lie outside that distribution around the parents. But still, this is a nice looking distribution with lots of variation to consider in genetic mapping. Richard found 4 quantitative trait loci for this contrast of two domesticus strains from different populations. And here's a plot showing the effects of these loci. So on the x-axis of each of these plots is the F2 genotype at a marker that's that's tightly linked to the QTL. And so GG means Gough homozygote, WW means WSB homozygote or mainland homozygote, and then we have the heterozygote GW. And you can see once again, remembering that the Gough parent had the higher recombination rate genome-wide. You can see that some of the QTL show that pattern of the high parent, the allele from the high recombination rate parent increasing recombination rate on chromosomes 5 and chromosome 6. But there are also QTL that show other patterns, including this one on chromosome 14, where the allele from the high parent is decreasing the total number of crossovers. This is again an antagonistic, a QTL with antagonistic effects that is similar to what Beth found on the X between subspecies, but this time it's on the autosomes. Okay, so Richard was also able to take this a step further. If you think about our experimental design here, when you extract spermatocytes from the testis, you can get a large number of spermatocytes. And that means that, you know, what I've shown you results for so far is mapping the mean number of crossovers in an individual. So you take 20 spermatocytes, for example, count the number of crossovers in each, and then take the mean, and that's the value for each individual. But because Richard measured recombination rate in each cell, he could also compute the variance for each individual, which raises the question of whether you can map QTL for within animal variance of the recombination rate. You might ask, why would we be interested in this? Well, the recombination rate is a cellular trait, right? When I talk about there being an individual recombination rate, it's always an amalgamation of a bunch of different cells. And so thinking about the variation among cells might tell us something about the process of how recombination rate is evolving. So here's the distribution on the left of that variance in the total number of crossovers. And you can see that Gough Island mice have on average less intercellular variance than WSB, the mainland strain. Interestingly, Richard was able to identify a single QTL that affects the variance. And in this case, it goes parallel mostly with the difference between the parents. So the Gough allele, which is coming from the parent that has lower intercellular variance, also is associated with lower intercellular variance in the F2s. But this is mostly just a sort of fun thing I wanted to show you and to help demonstrate how powerful this data set is. We can also go, particularly in this case where we're looking at differences between two domesticus strains, we can go deeper to identify candidate genes in these QTL intervals, because we have expectations for the type of gene that should show up. So for example, it not only needs to be expressed in the testis, but it should be expressed at the time or shortly before recombination is actually happening. And we can also look for differences, amino acid differences or potential upstream regulatory differences. And Richard used these filters to identify a suite of candidate genes. And I just want to show you one that is particularly, we think is particularly strong, although it needs to be tested functionally at this point. And that is RNF212, which functions is known to resolve double-strand breaks into crossovers. And some of you may know that there's been interesting work using the very large pedigrees in Iceland in humans that has associated RNF212 with natural variation in the recombination rate. So perhaps we're seeing something similar happening in mice, but we need to test this functionally before we can say for sure. Richard also is able to look at another piece of the pathway that leads to crossovers. You might remember I mentioned this thing called the synaptonemal complex, which functions as the glue for the homologs right before crossovers happen. And but before Richard came along, despite functional work and biochemical work on the synaptonemal complex, no one had ever attempted to map genetic determinants of synaptonemal complex properties in an evolutionary framework. And so what he did was he used an automated visual analysis to reduce these synaptonemal complexes from the microscopic images we have. Remember these red lines, what we're interested in, down to their main pieces. And then he developed a method to essentially estimate the lengths of these lines that took into account their curvature and sum them together to get an overall synaptonemal complex length for each cell and then for each mouse. And when he did that, he was able to once again map QTL, this time for the evolution of synaptonemal complex length. This has never been done before, and it was pretty exciting to us. He did it both in Beth's cross between the two subspecies and in his cross between two strains of Domesticus. And he was able to find several QTL, including on the X chromosome. But we were really interested in the synaptonemal complex because of its potential to impact or explain the evolution of recombination rate. And what Richard showed is that, in fact, for several of the QTL, he found QTL for SC length, the synaptonemal complex length, actually explained the QTL for MLH1 count using something called a mediation analysis. And so this suggests that one of the ways mice evolve more crossovers is by extending their synaptonemal complex, which gives them more real estate over which crossovers can occur. Okay, I just want to check real quick how I'm doing on this. Is good. I think I'm doing okay on time. Okay, so the next question I'd like to address is, does recombination rate evolve separately in females and males? There are lots of differences and similarities between the two sexes and how they accomplish crossovers. So this seems like a reasonable question to ask. April Peterson in the lab used the same immunocytology approach I mentioned to measure genome-wide crossover rates in a collection of inbred strains from the 3 subspecies I mentioned, plus one more hybrid subspecies that's called molossinus. And I'll just jump straight to the results. What I'm showing here is, so along the y-axis is the name of each inbred strain along with its subspecies or species. We have a couple of outgroups on the right side, Spreadis and Speciligus. And on the y-axis is again the average MLH1 foci per cell. So this is our measure of the total number of crossovers. The pink or orange circles are females, and the blue or turquoise triangles are males in all cases. And so for each strain, April was able to get an estimate of the genomic crossover rate or number in both males and females. Now, the reason this is important is that these are fully inbred strains. So the most of the genome is identical in males and females, with the exception of the X chromosome, where females have 2 X's instead of 1. So most of the genome is identical. And this allows us to this paired comparison gives us insights into how recombination rate evolves that we can interpret the differences without worrying about genetic variation among females and males that would show up in similar studies in outbred taxa like humans. Okay, so what we're really looking at is the relationship between female and male recombination rates in each strain. The first observation we can make by looking at this fascinating data set is that the relationship varies among strains. So the genetic background affects the relationship between the female and male recombination rate, which is an interesting result. Another perhaps surprising result is that relationship varies considerably to the point where the sex that has the higher number of crossovers varies. For example, if you look at the beginning on the left here, we have a couple of strains where the male rate is higher than the female rate, which is quite unusual in mammals. So we call this heterochiasmy. the difference between male and female rates, and you can see that heterochiasmy is itself varying. So the sexes appear to be evolving differently, at least based on this snapshot, when it comes to recombination rate, despite using the same pathways to accomplish crossovers and so forth. The biggest contrast that April found was in these rates among males. So if you look at the beginning strains here, you see there's just a huge amount of variation among males. And so she looked at her microscopic images to try to figure out what sorts of features high recombination males have. And first of all, the crossovers are distributed throughout the genome. They're not just on one or a few chromosomes, so it's a genome-wide effect. They also generate proportionally more double-strand breaks, suggesting that the elevation in recombination rate starts early in the meiotic pathway. And somewhat surprisingly to us, they also show stronger crossover interference, meaning that the spacing between crossovers, when you have two of them on a chromosome, is bigger or more regular than you would expect if they were happening at random. So that's a bit of a mystery, but that's what April found. She also found higher intercellular variance in females versus males, and she found this consistently across all strains. So this means that oocytes vary more in their overall crossover rate than spermatocytes do within animals. The circles are females, the triangles are once again males. And the fact that this is consistent across strains makes us wonder if maybe selection targeting recombination in a sex-specific manner might be more effective in males. That is, if the variance is lower in males, the intercellular variance, that means that the genotype of the phenotype of a mouse is a more consistent or robust readout of its genotype, which could increase the effectiveness of selection in males. We don't know, but that's just an idea. Okay, and then really quickly, to wrap up here, I just want to touch on this question of an evolutionary process that could shape recombination rate evolution. This is the most speculative part of my talk. The results are super fresh and new, so we'll see what you think. There are a whole bunch of models that have been developed to try to understand or predict how recombination rate should evolve. And I'm not going to subject you to all the details of these models, but I want to present one that seems to have some general appeal. I'm just going to give the logic and then we'll begin to test it. So the idea is that genetic drift generates associations between linked beneficial mutations. Sometimes this is called positive linkage disequilibrium. And when this happens, haplotypes containing the linked beneficial mutations tend to be fixed by positive selection because they don't have the deleterious mutations linked to them that will slow them down. These fixations in turn leave behind associations between linked beneficial and deleterious mutations, so-called negative linkage disequilibrium, that inhibit the response to selection. Okay, now what all this means is that if you have a mutation that increases the recombination rate, we would expect it to be favored, particularly in small populations where the effective drift is going to be high. Okay, so we're proposing, or people have proposed in formal models, actually, mathematical models, that this kind of standing association between beneficial and deleterious mutations, which is going to be greater in small populations, will select for increases in the recombination rate. So what does this mean? Well, we predict that in small populations, we should see higher recombination rates. all else being equal. And this prediction really hasn't been tested in nature. So here's how we're proposing to test it. We've sequenced genomes from population samples from a number of different populations. We estimate the affected population sizes of each population using demographic modeling. We also estimate the population recombination rate, which I'll say more about in a minute, called rho. And then we compare rho in populations with different affected population sizes. Okay, and again, the prediction is that it's going to be higher in, well, that the recombination rate is going to be higher in smaller populations. So far we've done this with 2 sets of mice, white-footed mice from islands in Boston Harbor, where our demographic models indicate a very strong reduction in the affected population size compared to a mainland population we've also sampled. And house mice, again, from Gough Island, where our modeling also indicates a strongly reduced affected population size compared to a mainland population. Let's see, am I going way over? No, I'm okay. All right, almost done here. Okay, so we need to keep in mind that these island populations are not probably at demographic equilibrium. They've experienced recent population bottlenecks. And most of the approaches that are analytical approaches that are available to estimate the population recombination rate, rho, on the fine scale, assume that populations are at a constant size for a long period of time. So we don't want to use those. But in 2019, Gustavo Barroso published this interesting method that tries to deal with this issue and jointly estimate along a chromosome values of the population recombination rate rho along with coalescent times that incorporate the effects of changes in the population size. Okay, so the way this method works is you have a single individual, a diploid, where you've sequenced its genome, and you walk along the chromosome, and at each point along the chromosome, you can estimate rho and also estimate the time to the most recent common ancestor for the pair of chromosomes, the pair of homologs. And you end up getting this kind of map of rho along the chromosome in a way that is supposed to account for changes in the affected population size. Now, it's important to keep in mind that even accounting for that, what you're estimating still has the affected population size in it, okay? So rho is equal for an autosomal locus to 4 times the affected population size times C, which is the recombination rate we're interested in. But I'll talk about that in a second. So we get these rho maps, and this is what it looks like. This is very busy. I don't expect you to dig into this too much, but what we're showing on the y-axis for each chromosome is the average rho, the population recombination rate in 1 megabase windows along the chromosome. And the dotted lines represent the average across the chromosome for each population, and they're color-coded on the right here, okay? There's the legend. And each line, the fluctuating lines, are each coming from an individual, because again, we can measure rho using each individual and then take an average, which is the dotted line. So here we're showing the first 6 chromosomes for white-footed mice on islands. And what I want you to see is that the average recombination rate, the average rho, the population recombination rate, is, according to this method, lower in 2 island populations than it is in the mainland. Okay, it's lower. Now remember, rho is 4Ne times the recombination rate. So some of this difference may be because Ne in the island populations is actually lower. But let's look at another data set and see what else we find. In house mice from Gough Island, compared to our mainland population of Germany, these German sequences were collected by Bettina Haar. What we actually see is the opposite pattern, where there's a higher row in Gough Island mice. And we've done statistical comparisons, and it's actually significantly higher in most intervals across the genome. You can also, by the way, see in these plots that there's a lot of heterogeneity in the recombination rate, indicating that there may be hotspots of recombination that we know to exist in house mice. But anyway, white-footed mice so far are showing a lower row in islands, whereas house mice are showing a higher row on Gough Island. So this higher row, because we expect the Gough Island mice to actually have a lower affected population size, the higher value of row is going in the, can't be explained simply by affected population size being part of row. We think that this result provides preliminary support for the prediction that the recombination rate is higher in a population with a lower effective size. But it's very speculative at this point, and we're just beginning down this path. So I will summarize and give you some conclusions across these three questions I've mentioned now. We have concluded that based on our genetic mapping, that recombination rate looks a lot like a standard quantitative trait in that its evolution is conferred by an architecture with moderate genetic complexity. There are multiple loci with modest effect sizes in general, although we have this big one on the X. And we can also see that the QTL, the regions of the genome that are involved in conferring changes to the recombination rate, are different between and within subspecies. Another more subtle conclusion that is, I think, kind of interesting is the fact that some of the QTL are showing antagonistic effects, suggests that the differences in recombination rate we see between populations and between subspecies are not just the result of directional selection going towards a sort of constant optimum, right? That would predict, probably wouldn't predict to see these antagonistic effects in that scenario. We see a role for the synaptonemal complex, which merits its study in an evolutionary context. One of the more exciting and puzzling results for us is that the males and females have what appears to be distinct genetic and evolutionary trajectories, which suggests that there are contrasting selective pressures in females versus males when it comes to the recombination rate. And a very tentative thought I ended with is that selection may favor increases in small populations, increases in the recombination rate. And we're about to do a whole bunch of simulations and look at other populations to see if we can arrive at a firmer conclusion there. Okay, the last thing I'd like to do is just say thank you to all of you for listening. I'd like to thank the university for all of its support, and the National Institutes of Health have been quite generous in keeping our recombination studies going. And this is just a picture of my some members of my lab and a reading group that we do. I want to let you know, since a lot of the top folks in population genetics reside in Vienna, that I'm interested in recruiting postdocs, particularly people who are interested in working on recombination or the island rule, and people who are excited about house mice and how they might tell us something new about the process of evolution. So if you're interested, please get in touch with me. Thank you very much.